Hi, welcome to the Bioinformatics Chat. My guest today is Simeon Carstens, and we will be talking about Bayesian analysis of high-C data. Simeon, welcome to the podcast.
Hi.
Simeon, my understanding is that you're a physicist. So how did you transition into bioinformatics and biology and biophysics?
Right. So up until my master's in physics, I pretty much focused on theory and in the end did my master's thesis in a subject in computational astrophysics.
Astrophysics?
Yeah.
Wow.
Exactly.
This is one of the, most unusual profiles of people who have been on this podcast.
Right. Yeah, it was quite a change getting into biology then. But why I did this eventually was that I had the feeling, while astrophysics is interesting, it's a very remote thing, and kind of wanted to apply the computation methods I've been using for my astrophysics subject in a more life science oriented research field. And then I was on I looked for positions and found a sort of algorithmically oriented group, which I found a good match because it allowed me to focus on the methods and not necessarily dig too deep into the biology, although of course I still had this option.
So that was for your PhD?
Yeah, exactly. So I started my PhD right after my master's in physics, yeah. And for my PhD, I changed to structural bioinformatics, if you want, yeah.
Right, and you just completed your postdoc, right?
Yeah, that was my second postdoc.
Were you working exclusively on high-C, or what other areas of biology did you touch?
So in terms of biology, the data I worked with was exclusively high-C. Another part of my PhD was about Markov chain Monte Carlo sampling methods, so very algorithmically oriented. They are useful in general for Bayesian data analysis, which we might get into later. But in terms of applications of those methods to actual biological data, I exclusively worked on high-C data.
And we should also mention that now that you finished your second postdoc, you're looking for a position, and not in academia, but in the industry. So, if any of the people listening to this podcast, if you have any openings, and you'll get a sample of Simeon's competency as we go along here, but definitely get in touch with him.
That would be amazing. Great.
Let's start with learning about high-C. I imagine a lot of people listening to this either have never heard of high-C or maybe heard, but don't really have too much experience with it. So what is high-C?
So high-C is the, let's say, most recent incarnation of a class of methods which allow to query the three-dimensional structure of chromatin. So this class of method is called chromosome conformation capture, or for short 3C, and was, I guess, invented when was that? 2002, perhaps? And in the beginning, you could query two single loci. Basically, you would get a contact frequency between two single loci.
So let's clarify this. Maybe let's start with what is chromatin for those who maybe don't remember.
Yeah, so chromatin would be the functional form of DNA, most in the interface nucleus. So basically, it's DNA with associated proteins, mostly histones. Depending on which website or which definition of chromatin you look up, also RNA is counted towards chromatin. But it's basically the stuff, functional, or it's the functional form of DNA in the interface nucleus.
Right. So is the way the DNA appears in the cell, or is packaged in the cell and the nucleus.
Right. Exactly. So it's not the naked DNA, but it's sort of pre-organized on those histone proteins, and this complex is called chromatin then.
Why exactly are we interested in its 3D structure?
This is also a recent thing. I mean, from imaging experiments, which were already there before, the high-C experiments, obviously, you had information about, say, chromosome territories, and you were able to label single genes and see basically where they are located in the nucleus. But then it turns out that similar basically to a protein, the sort of fold of DNA in the interface nucleus is of functional relevance. So the 3D organization of chromatin is a epigenetic regulation or gene regulation mechanism. And this makes it obvious that the 3D organization of the genome is crucial for many biological processes.
Right. And so this method called 3C was invented, and 3C sort of stands for, is like a tongue-in-cheek way of saying CCC, right? Which stands for chromosome conformation capture. And what was that method like, and what were its limitations?
So the basic concept is that you cross-link cells, you fix them using formaldehyde usually. Then you would add a restriction enzyme to digest DNA at the predefined restriction sites. And then you would ligate those dangling ends you basically have of these restriction fragment ends. And using those ligations, you get sort of a little, if you want, chimeric sequence. I don't know whether that's the correct expression, but imagine two loci on a chromosome which are far in sequence but close in space. After fixating the cells and ligating, you would have a short chimeric sequence of those two loci. And this ligation junction can then be detected. That way you get information about which loci have been close in space at the time of cross-linking.
Right. So that was called 3C, right?
Yeah, that's the basic process in all these 3C-based methods. And the very first 3C-based method, called 3C, was able to query only 2 select loci, basically. That was the crucial limitation of the very first incarnation of this method.
And then there were other methods like 4C. What was there also 5C as well?
Yeah, exactly. So basically those methods, 4C, 5C, subsequently high C, increasingly yielded better coverage. So 4C is a one versus many method. So you would be able to query one locus against a larger set of loci. While in 3C you could query one locus against one locus. 5C then would be a many versus many method. Basically you query many loci against many other loci, thus allowing you to investigate the 3D organization of a whole genomic region, basically. And high C then is the ultimate coverage, which is genome-wide, all versus all.
I guess nowadays we just use high C. Is there any point in doing those older methods or previous methods?
Oh yeah, absolutely. It depends on the application you have. So one drawback of high C is that it involves a lot of next generation sequencing with the associated costs. And if you're only interested in say a smaller genomic region, you might as well just take 5C, which is also sequencing based. Or if you're interested only in how one locus interacts with several other loci, you might as well pick 4C. And that way save money and might have an easier protocol to follow, actually.
Interesting. Okay, so once we've done one of these experiments and the data we get for those higher C things where we don't need to like explicitly design primers, we get sequencing data as with DNA sequencing or RNA sequencing, and unlike other sequencing experiments, we're interested in very specific sort of reads, the reads that indicate a junction between two genomic pieces that come from very different positions on the chromosome. So we're looking for those events, and that sort of tells us about which parts of the chromosome were close to each other in the 3 dimensions.
Absolutely. Not only of one chromosome, but of the whole genome. You might have basically interchromosomal contacts as well. Yeah.
How do you analyze this data? So let's say you filtered your read set and you found those interesting reads. How do you then summarize them to make sense of them?
Right. So the first step, once you do have your sequencing data, would be to align those reads to the reference genome of the organism you're considering, which then actually allows you to associate the loci you detected with specific loci on specific chromosomes. This gives you the information which loci has been in contact with which other loci, how many times. Yeah. And the data you eventually get would be a contact matrix. So imagine a single cell case. You would have a matrix on the axis. You would have the sort of chromosomal length in base pairs. And you would get a dot in this matrix if you measured one of those ligation junctions at the loci corresponding to this dot, basically, which are indicated by the X and Y values. And this matrix then would have a strong diagonal. It's easy to imagine because a dot on the diagonal means that two loci, which are close in sequence, have also been close in space. And that does make totally sense.
Right, so it would be something like a small loop in the DNA or some kind of twist.
Right. I mean, the loops are then the interesting contacts, right, which are off the diagonal because they are farther away in sequence, but still close in space.
Right. So when you were talking about the strong diagonal, did you mean things that are not really even ligation events?
I guess you would try to filter those out before you get ligation events close to the diagonal, but they don't really contain that much information, right? Because as DNA is a polymer, it's obvious that loci which are close in sequence are also close in space. So the counts close to the diagonal would be much less informative than the off diagonal.
Exactly.
Counts.
So because you can tell from the sequencing data whether there is a, chimeric read, whether there's a ligation event there or not, or if it's just contiguous.
Actually, you can, because you selectively pull down those fragments which do have a ligation junction. So those fragments are marked with biotin, and you can use this to selectively pull down actually those little chimeric sequences.
Oh, that's great. Right. So if you are considering just the ligation events, why are you still seeing the strong main diagonal in this matrix? Is it because there are like small twists in the DNA, or why is that?
Yeah, absolutely. You could have that's probably what you meant before. You can have very tiny loops, right, of between loci which are close in sequence. Yeah. There's actually very interesting work on that, which uses this information of ligations which are very close in sequence to determine bounds for the bending stiffness of chromatin. This is very interesting work because obviously, if those loci are too close and the DNA has a certain stiffness, you can't bend it so that you can ligate those free ends.
So you get this contact matrix, and just by looking at it, so you could visualize it as a sort of a heat map where it's higher temperature, the more contact points you got there, and presumably you use some binning, right? So you consider like intervals on the chromosome. So you use some binning, and you visualize it as a heat map. So what can you tell from a typical contact map or contact matrix just by looking at it?
Right. So one has to be clear that before binning, in the single cell case, speaking about a heat map doesn't really make sense, because between two specific loci, in the single cell case, you can have a maximum of 2 contacts for loci on chromosomes of which you have 2 copies in the cell. So in the ideal case, where there's no like half replicated chromosome or something, your maximum count in this matrix would be 2. So I wouldn't speak of a heat map yet in the single cell case. On the other hand, this experiment usually is performed on whole cell populations. And then you're right, a heat map would give you a contact frequency or something most likely proportional to a contact probability. So that's an important distinction to make, I think.
Yeah, absolutely. And I think we'll talk about the single cell case a bit later. I was assuming that the bulk high C is sort of the normal case as it was in like RNA sequencing. That single cell is a more late development.
Right.
So usually you would see in that heat map, you would see some kind of structure, right? There are these things that are called TADs. Can you talk maybe a bit about those?
Yeah, absolutely. So if you look at the pre-processed and filtered heat maps, you see a sort of plate pattern or blocks in this matrix. And those correspond to different levels of sort of domain organization in the DNA, if you want. So the biggest blocks have been found in the very first Hi-C publication and would be called AB compartments, which are alternatingly active and passive in terms of transcriptional activity. And within those AB compartments, you would have sub-blocks, like little beads on a string, which would then be the topologically associating domains or TADs, which you just mentioned. Yeah, this is currently very much under investigation, whether those TADs are present in each single cell, whether they're an averaging effect. But they do seem to have the property of being a fundamental structural organization element. So to the best of my knowledge, TADs are conserved between tissue types, for example, but not necessary between organisms.
Interesting. So these TADs, these domains, they're sort of analogous to protein domains, which are like consistent substructures within a bigger protein. So if you consider chromatin as something like a protein, then these domains, I guess, are in some way analogous?
In a sense, yeah. You can actually model chromatin as a beads on a string model. That is what we do in our research, and that's also what other groups do. Other groups actually use the information about TADs to build those beads for their modeling procedures.
Mm-hmm. So TADs play the role of the beads on a string in this model.
In that specific model of that group, yes. Yeah.
Okay, and you also mentioned the single cell Hi-C, which sort of follows the general trend of making everything single cell. What kind of experimental developments enabled, performing Hi-C in single cells? Is it more or less analogous to single cell RNA sequencing, or is it very different?
So to be honest, I'm not too familiar with single cell RNA sequencing. What I can tell you is that the first single cell Hi-C paper came out in 2013, which is I think 3 or 4 years after the original Hi-C publication, and it actually involved picking single nuclei under the microscope and then performing an adapted Hi-C protocol on the single nuclei. single-cell high-C has been developed further by increasing throughput. So in the meanwhile, you can actually have several thousand of single-cell high-C maps from one experiment.
Speaking of the single-cell high-C, as you said, there's not enough data to make heat maps really, because it's either on or off. So it's a very particular kind of data, right? So how do people analyze high-C data, and what inferences can you make out of it?
So what people often do once they get a high-C matrix, be it single cell or population high-C, is detecting those TATs, for example. So there are a lot of methods to call those TATs. And we just talked about these block structures, which you immediately notice just by looking at one of those matrices. But very interesting are also single points of higher contact frequency, which are off the diagonal, which would correspond to basically attachment points of loops. We would have increased contact frequency. So this is an important thing people look out for, because they might correspond to, say, a promoter-enhancer contact. So
is this in the context of single-cell high-C? Or are we talking about bulk now? Because I'm thinking if it's single-cell high-C, right, then if there is a single point of contact, wouldn't it correspond to roughly like a single dot in the matrix?
Yeah, absolutely. So that last comment about the points of increased frequency of the diagonal, that definitely applies only to population high-C, as the single-cell high-C data is way too sparse for that.
Right.
Another interesting thing you can deduce from the contact matrices alone are how strongly the contact probability between two loci decreases with the sequential distance, which tells you something about the polymer physics of chromatin. This contact probability as a function of linear distance is a measure predicted by different statistical physics polymer models for DNA. And depending on the assumptions which go into these models, this might also tell you again about how, for example, the dynamics, or how chromatin was getting into this organized form it now is in the cell.
And so in one of your previous papers, you introduced a specific modeling framework for single-cell high-C data. And can you talk about that?
Yeah, so the modeling framework we introduced is based on an approach my PhD supervisor developed called Inferential Structure Determination. And before going into detail about the high-C modeling, I should probably quickly explain what the original approach is about. Inferential Structure Determination is a Bayesian view on structure determination from any kind of biological data. So what you would traditionally do is you would take a set of, or you would calculate many trial structures, back calculate experimental data from them, and make this back calculated data fit to the experimental one to basically find structures which reproduce your data well. And you augment this with physical a priori information about, say, a force field between amino acids, or connectivity, which enforces the restraint that we deal with polymer chains. And you would find a optimal structure which fits the data best, and also the prior information.
Right. So we should clarify that your goal here is very ambitious. It's to actually reconstruct the full 3D structure, because a lot of times I think when biologists do high-C experiments, they're mostly interested in like what lies close to what, just to predict where maybe enhancers are located or something like that. Mm-hmm. But as a biophysicist, you're not content with that. You want like the real thing.
Right. Yeah. Yeah, that's indeed what we're doing. We are trying to find a three-dimensional, well, at least approximation to the actual structure, which will, of course, be coarse-grained, because we can't model each base pair separately. Now, the approach I just mentioned, the classical one, basically fitting back-calculated data to experimental data using some optimization method to find the best fitting structure, is, in our view, kind of problematic. So one thing is you usually end up with one optimal structure, and you have no measure of certainty about the structure, right? If you have a minimization problem and your algorithm is, does what it's supposed to do, you will always end up with with very similar structures which correspond to the global minimum of your scoring function. But you don't have any information about how broad basically this sort of basin is in which this optimal structure is located. And furthermore, these approaches lack any clear statistical foundation. So it's very hard to speak about, say, an error bar for your structure, about a structural standard deviation. And another thing is that many of these approaches require the a priori setting of modeling parameters, which are not of primary interest. And you would set those to sort of arbitrary values, but it's really not a very objective process. And ISD tries to improve on all those aspects.
What was the context in which the ISD was originally introduced?
Yeah, so ISD was originally introduced for protein structure determination from NMR data.
And so we already mentioned the rough analogy between protein structure modeling and chromatin structure modeling. So it sounds natural to apply the methods from proteins to chromatin. And that's what you did, right? So what does your model look like?
So we actually proposed several models. So much of the NMR data, which was used to model protein structures back then when ISD was invented, was basically distance data. You would have distance restraints between different amino acids in this case. And we can sort of transfer this model to Hi-C. So it's sort of common practice to derive distance restraints from Hi-C data. So basically from the experimental data, you propose a model which tells you that given this data, the distance between two loci, in the case of Hi-C, is supposed to be that and that or within a certain range. And you would use this information, which comes from your modeling assumptions, as a restraint, meaning that your structure which you're optimizing, or the real structure which you're looking for, is supposed to fulfill this restraint. So in the actual structure in your model, two loci should fulfill this restraint, be within that range or be approximately that certain distance apart.
What exactly are the assumptions that lead to restraining the distance? Could you give a specific example?
Yeah, so these assumptions most often come from polymer physics. So it's of course a very approximative thing. So in population Hi-C data, you would of course expect that if you have many counts between two loci, then on average, those two loci would be closer in space as compared to two loci which have few counts in the Hi-C matrix. And so you would have a sort of inverse relation between the distance or the proposed distance and the Hi-C count.
Okay, so this restraint function comes from the data, right? It's not a priori based on your model, but it incorporates the data as well.
Exactly. It does incorporate the data, but the specific form of the restraint function is a somewhat arbitrary choice. You can motivate it partially from polymer physics, But then again, you're never sure how appropriate your polymer physics, or your polymer physics model, which leads to this special, or to that specific form of this relation. You're never sure how appropriate this actually is, given the little information we have about polymer physics of chromatin.
Right. So the distance restraint tells you how well your 3D model corresponds to the data. Is that roughly correct?
That's absolutely correct. Under the assumptions of the model you're choosing. Yeah.
I guess this is compatible with both the Bayesian approach and the simple sort of optimization maximum likelihood approach. So where does the Bayesian magic come in?
You're absolutely right. This is shared between both approaches. The Bayesian magic comes in by asking the question in a different way. The question asked in Bayesian approaches, or specifically in inferential structure determination, is what do we know about a structure given data and a priori information? And it turns out that this question is being answered by the posterior probability distribution over structures. So basically, for a given structure, you would get some value between 0 and 1. 0 meaning you're absolutely sure this structure is not the correct structure under the assumptions of your model. And the value of 1 for this posterior probability being you're absolutely sure that this structure is the correct structure.
But then I assume your structures populate some kind of continuous space. It's more like a probability density there.
Yeah, it is. Absolutely, yeah.
So it could get out of the 0-1 interval.
Right, yeah. Yeah, sure.
And how exactly do you model that space? So in order to perform either optimization or Bayesian inference, you have to specify the space of all possible conformations. So you already mentioned the beats on the string model.
Mm-hmm.
And walk us through the specifics of how that model is set up. Like, for example, how many beats do you have? What do they correspond to?
Right. So you mentioned before that you would usually, before doing structure modeling, you would bin your high C matrix. So those bins would then correspond to beats on a string. So basically, we approximate the chromatin polymer by sort of a beats on a string model, where we have spherical units linked to each other, and each spherical unit, called a beat in this case, corresponds to a certain amount of DNA. And we approximate, this stretch of DNA contained within those units as a spherical thing, basically. Now there's, in our model, two components to this polymer model. One being a volume exclusion term, which makes sure that that two beats don't overlap too much. Otherwise, you would tend to get very compact structures. And the other component is a sort of bond length restraint, right? So if you have two neighboring beats in this beats on a string model, in this chain, you don't want them to be further apart than a certain distance to avoid that you get a very stretched out polymer.
So that's the role of the string, right? The string doesn't let the beats get too far apart from each other. And the first condition means just that your beats are rigid or at least almost rigid?
Almost rigid, yeah. That is a parameter actually, which is sort of free, and we have to set a priori. So honestly speaking, also our Bayesian approaches are not free of parameters you have to set in advance. And this is one of those parameters. Yeah.
That's the dirty secret of Bayesian modeling.
Yeah, so to say, yeah. I mean, but the thing about Bayesian modeling is that you're always explicit about your assumptions. So in a sense, it's still more honest than traditional approaches, I think.
Right. And after you specify this model, your beads and your string, you have this issue that the same 3D model could be rotated arbitrarily. So if you just specify a structure by the coordinates, by the 3D coordinates of each of the beats, then to each structure there is an infinite number of ways to specify it, accounting for all the rotations. And this poses a problem for Bayesian inference. So it is very well known in the case of mixture models where you have multiple modes, but at least there we have a finite number of modes. But here there is an infinite number of modes. So how do you deal with that? How do you sort of normalize this to a, so that you have just a single way to specify a single structure?
Right. So one thing you can do is choose different coordinates. So in the original ISD formulation, the original ISD work on NMR structure determination, protein would be actually parameterized by internal coordinates, so basically by bond length and torsion angles. So this is one way of getting rid of these additional degree of freedoms.
So you fix one of the amino acids as sort of the origin? Is that the idea?
Yeah, absolutely.
Right, but how then... still you have to specify the orientation. So do you have to fix like 2 amino acids to specify to account for the rotational symmetries?
Yes, yeah, true. But in our case, we actually do use Cartesian coordinates. But it turns out that if you rotate your structure, of course it's the same thing, but it also has the same posterior probability. So to be honest, I don't really see the problem there. I mean, the posterior distribution is then sort of invariant under these rotations or translations, right?
For sure. I think it's just a matter of efficiency because even if you're trying to explore the space of sort of the highest probability density, you already have an infinite space to jump around, and that's even before you get to some of those less probable, volumes of your space.
Right.
So I think it just becomes much less efficient to sample the space with a lot of symmetries in it and many modes.
That is true, but in reality, actually, it turns out that so we use Markov chain Monte Carlo methods to sample from the posterior distribution. And in the Markov chain, the subsequent states are to some extent correlated, right? So state I would have a very similar orientation to state I+1 usually. Mm-hmm. But nevertheless, it's still correct that once we get our final ensemble of structures to compare them, we would have to align those structures. That's absolutely right.
Right. And maybe let's say a couple of words about Markov chain Monte Carlo. And it's funny because this is, I think, this is going to be the third podcast in a row where we mention the name of Andrei Markov. Because we talked about Markov models and then hidden Markov models. Yeah. And now we have Markov chains, which are roughly the same thing, but in a completely different context. Right. So, so the main idea of Markov chain Monte Carlo is that, you want to have this Bayesian inference. You want to draw samples from your posterior distribution. And the idea there is that you set up a Markov chain, so a Markov model where you jump around your state space. So in this case, you jump from one conformation to another according to some rules, according to some transition probabilities. And the idea is that if you jump long enough and the states that you've visited will roughly, sort of their distribution will roughly correspond to your posterior distribution.
Yeah, absolutely.
And there are different approaches to Markov chain Monte Carlo. So which specific methods are you using in your method?
Yeah. So two things. One thing I might just interject for completeness here is that we're not only sampling the structures, but also nuisance parameters, which are model parameters which are of secondary interest only. So a state in our Markov chain would not only be a structure, but also a set of nuisance parameters. But that's just a note.
What, for example, are those nuisance parameters?
Right. So it's actually a very important topic. In the likelihood you introduce, which basically describes sort of the goodness or fit between your data and the back-calculated data, you would allow some deviation between, or some difference between those two, because of course experimental data is noisy and your model might not be perfect. So you allow some kind of error. You could, for example, use a Gaussian error, which then introduces the standard deviation of the Gaussian distribution as a modeling parameter, which is not of primary interest because we're interested in the structures and nothing else, actually. But still, we have this noise parameter, which we try to infer from the data, and which in previous approaches was set to heuristic values or determined by cross-validation.
Yeah, that makes sense.
So we have to realize that the parameter space is very large we're sampling. So if we have, say, a typical number of beats for one chromosome in our first single-cell based work would be, say, 300 or 330 beats. And each beat has 3 Cartesian coordinates. So we would have a sort of 1000 dimensional parameter space. And because of the data and the force field, the beats on the string model, all those coordinates are very highly correlated. So you can imagine that the posterior distribution is very complex, definitely of non-standard form, and multimodal as well, and high dimensional, of course. So for these reasons, we need pretty efficient and advanced Markov chain Monte Carlo methods. And we actually use a combination of those. The most important method we use, I would say, is hybrid or Hamiltonian Monte Carlo, which actually exploits the geometry of the parameter space. Or, well, that's maybe a little inaccurate. The sort of gradient information of the posterior distribution to propose better states, which are then accepted or rejected using a metropolis criterion. Yeah, so that would be hybrid Monte Carlo, again, using the gradient information of the posterior distribution.
So hybrid here refers to the hybrid of Hamiltonian and Markov chain, right? Because Hamiltonian, I think, is considered like separate from Markov chain Monte Carlo.
I think hybrid more refers rather to the combination of Monte Carlo or metropolis Monte Carlo and molecular dynamics. Because to get these proposal states for your Markov chain, you actually simulate a very short microcanonical molecular dynamics trajectory that you can imagine the way that your parameters represent a point particle which moves under the influence of a force. And that force is given by the gradient of the log probability of the posterior distribution. And you basically integrate the equations of motions for this particle for a certain number of steps. And the endpoint of this short molecular dynamics trajectory would then be your proposal state for the next Markov chain state.
Right. So that's the Hamiltonian, right? Monte Carlo. Mm-hmm. And then but you also mix that with some more sort of Markov chain methods like Gibbs sampling, I think.
Absolutely, exactly, yeah. So depending on how you choose your statistical models for the prior distributions and the likelihood, turns out that the conditional distributions for the nuisance parameter or parameters are often of standard form. So you would often have a, say, gamma distribution, for example. And we then exploit Gibbs sampling to split up the difficult task of sampling from the joint distribution of structures and nuisance parameters into, iteratively sampling from the respective conditional distributions. So we can make use of already implemented random number generators for those standard distributions. And then use Hamiltonian Monte Carlo only for these structural parameters.
Mm-hmm. And how do you validate this model, like both your inference algorithm and your generative model in the first place? How do you make sure it actually makes some physical sense? Like, can you take a posterior distribution, the samples from your posterior distribution, and somehow, sanity check it?
If you don't mind, I'd like to mention one more important Markov chain Monte Carlo algorithm we actually use.
Sure.
So maybe we can come back to that question, which is an important one, obviously. So the point is that even using Hamiltonian Monte Carlo and the Gibbs sampler, the Gibbs sampling scheme can get stuck in local minima of the high-dimensional posterior probability distribution. That is why we use a technique called replica exchange, which is pretty popular, I think, in computational physics and in the molecular dynamics community. It basically works by not only simulating the posterior distribution, but in a sort of physics sense, also distributions or versions of it at a higher temperature, which effectively flattens the posterior distributions. And we would basically simulate up to several hundreds of intermediate distributions. So basically we have a chain between a chain of interpolating distributions, one end being the posterior distribution, and the other end being a very, very flattened version of it, which is easy to sample for the Gibbs sampler. And occasionally, this replica exchange algorithm would exchange states between neighboring simulations in this chain. And by this diffusive process, you can help the simulation in the posterior distribution to escape from local minima.
That's interesting.
That's a very important technique we use. Yeah.
So if I understand correctly, raising the temperature basically makes everything more chaotic and makes it easier to escape the stable state of...
Exactly. Yeah.
And this is why you say it becomes flatter, so
yeah,
even the weirder conformations become more probable, so you can jump into them with higher probability.
Yes, exactly.
This also reminds me, I don't know if you're familiar with this, I think the algorithm is called simulated annealing from discrete optimization. Is that roughly the same idea?
Yeah, that's a very similar idea, with the key difference that simulated annealing is a global optimization algorithm, right?
Yeah.
So the goal of simulated annealing is not to produce samples from a probability distribution, but to find an extremum of some scoring function. Yeah, but the idea is very similar. You have your scoring function at different temperatures, and you occasionally, you exploit this to you increasingly lower the temperature until you arrive at your target scoring function, and thus increasing sampling.
Right, and so we were also talking about model validation, right? How do you validate your posterior?
Right. So one thing we check is whether we can check the components of the posterior distribution separately. So we might, for example, check whether the assumptions of the force field of the structural prior model are fulfilled. Do we have many, say, overlaps or collisions between beats? And we, of course, also check how well we reproduce the experimental data. So a standard plot we do is a scatter plot between the experimental data and the back-calculated one for a representative sample from the posterior distribution. Those would be pretty naive checks, I guess. And then, obviously, and this is also very important in papers to convince people that our structures make sense, is we try to use independent experimental data to check whether our structures agree with them. So for example, we might use fish distances between certain loci and check whether in our model those distances are similar.
Right, where fish refers to fluorescent imaging, right?
Exactly, yeah.
But that still checks your most probable point in the space. So you're essentially checking that those confirmations that are most probable, they agree with the experimental data. But I imagine I could easily pass that check if my algorithm just found the optimal point and replicated it 1,000 times and would say this is my posterior. I would presumably pass that check as well. But
right,
how do you check that your posterior as a whole is sort of well calibrated? Can you check that?
Yeah, it turns out that, and that is one part of our most recent paper actually, that you have high throughput fish data. So you don't get a single distance, but you get a whole distance distribution for the distance between two specific loci. And with the structures we obtain, we can basically, plot a histogram of this distribution and see whether not only the mean of those histograms, basically the experimental one, and the histograms obtained from our models, whether not only the mean matches, but also the, say, spread of the distribution. And it turns out that works quite nicely.
Oh, that's pretty cool. And so whereas your first paper, or I don't, maybe not the first, but your previous paper on this subject was about applying the ISD, the Inferential Structure Determination, to single-cell Hi-C data. Your later paper, which is now a preprint, is concerned with the aggregated population Hi-C data. What is the difference? What are the unique challenges that are posed by the aggregated data?
Right. So it's actually a very interesting problem. So again, so for single cell data, your contact matrices are very sparse, right? So basically those matrices are almost empty. But the contacts you have correspond to, contacts measured from a maximum of 2 molecules, right? So for single cell data, you have very sparse matrices, but the contacts you get are very easy to interpret and subsequently also to model. While population-based data, the matrix is densely filled, but each data point is basically an average between an average of millions of cells, of millions of different genome structures. And that poses a challenge to reconstruct structures which reproduce these averages. Also in light of the question of cell-to-cell variability, right? I mean, can we safely assume that genome structures are comparable between different cells? That is a question which is very much debated, and of course, with the advent of single cell techniques, also being increasingly answered. And turns also out that one important result of the very first single cell Hi-C publication is that indeed the measured contacts are very different from one cell to the other. So in my most recent work, we basically propose a multi-state model. So one sample in the Markov chain would not consist of one structure plus Newton's parameters, but of a whole set of structures, say 20, 40, up to 100 structures, which on average reproduce the Hi-C contact matrix, the population-based Hi-C contact matrix. So it's basically demixing the average data to obtain a set of structures which reproduces it.
Right. So whereas at first your model space or your state space in which you were doing Bayesian inference, right? The space of your conformations, it was basically just a space of a single conformation, and it still was like 1,000-dimensional. But
yes,
now this is multiplied many times over, and you have this product space where you have many copies of that space, and you have to do inference in this huge space, right? That must be very challenging technically. How did you manage to do that?
Yeah, that's a sort of so basically putting the finger right in. It's a big challenge, and because it's such a difficult sampling problem, we had to limit our attention to a small genomic region, which comprises only 2 topologically associating domains. We actually use the same techniques, but with an increased computational effort. So just to give you some numbers, we would use in this replica exchange algorithm, I would use up to 300 to 400 replicas, and to get, say, 50,000 samples in the Markov chain, that takes 48 hours on up to 400 processes. So it's very slow, unfortunately. And that is, honestly speaking, also a sort of weak point of our approach so far, that at the moment with the current implementation, it's not scalable to, say, whole genome calculations at reasonable resolution. And we only regard a small genomic region of 2 neighboring TATs, but model that region by around 300 beats.
Right. So at the moment, it's more like a proof of concept that this is potentially doable, albeit on a small scale. But how do you choose the number? So the number of copies of the state space, which is the ensemble size, right, is basically you're saying, is this number of like maybe tissues or this number of different modes of chromatin that exist in this population, right? And
right.
How do you pick that number?
Yeah, that's a very interesting question. So in previous approaches, there was very little objective methodology to pick that number. But we actually treat this number again as a parameter which we don't set a priori, but which we want to infer from the data. So we make use of Bayesian model comparison as a framework. Basically meaning that we calculate or we simulate with the different numbers of states of conformers from 1 up to 100 in certain steps. Then we would calculate the evidence, which is basically the normalization constant of the possible distribution, and the likelihood of generating the experimental data under the assumption of our model. And the simulation, which results in the highest evidence, would then be preferred by the data. And that would be the simulation or the number of conformers we pick as the right one, again, under the assumptions of our statistical model.
And were you able to correlate that number inferred by your model to some sort of characteristic of your data, like, again, the number of tissues or something like that? Or like what was the number like in your analysis?
Yeah, so this number is at the moment, I have to admit, doesn't really relate to a biological thing. That is because our model is still very coarse-grained, and of course, both the likelihood to back-calculate data and the physical a priori model is just very approximate. So it's more a guideline on how to set an important modeling parameter. But in fact, we envision, and that would be actually very cool, that given more accurate models and fast implementation and just faster computers in the next few years, you could actually use these evidence calculations as a tool to measure cell-to-cell variability, right? I mean, imagine you have 2 populations of samples from, for example, different tissues, and you would expect, this is just an uneducated guess, I'm not a biologist, that, for example, a sample from cancer cells would be much more diverse because it's a very chaotic thing going on than, say, I don't know, a non-cancerous cell type. And then we would hope that simulations from cancer cells would give a higher number of states in the model as calculated by the Bayesian model comparison as compared to simulations from data from non-cancerous cells. But this is a very, very ambitious goal. And we don't say that we can do this yet, but at the moment it's just an objective way to set a modeling parameter.
And maybe this is a naive take, but I was curious if you could use your former model that you set up for the single cell high C to whether you could apply that module to population high C in the sense that it would be essentially a mixture, right? So you would collapse all these copies of the state space with their respective probabilities with which they occur in the wild in your sample. And the different conformations that occur in your sample would just become different modes in your posterior distribution. Would that work?
This is a very questionable approach. So one of the main motivations we actually came up with the multi-state modeling is that still most published methods to model structures from population IC data actually calculate consensus structures. So try to find a single structure, a single conformation of the chromatin polymer, which explains the data as best as possible. But again, in the light of cell-to-cell reliability, we think this approach is a very dangerous road to take. And then it's also important to note that if we get different modes in the posterior distribution, this is just a measure of uncertainty and not necessarily of structural variability, I think.
But those are very similar things, aren't they? So to be clear, I agree with your take on the consensus structure, right? But once you move into the Bayesian modeling, and once you have a posterior distribution, why couldn't that take care of the different things? So if you pose this question as, what is the distribution of the conformation? And the distribution is just a mixture of multiple distributions, and you currently try to model the multiple distributions, but you can also collapse them with their respective probabilities, and maybe this could be, this should definitely be more efficient.
Yeah, I see where you're going. So one thing we actually noticed in the multi-state models we calculated is that those are very diverse, right? So we actually try to cluster or find clusters of, say, dominant states or structures from our multi-state models, but that actually didn't work out. So if you go from one multi-state model to the next one in the chain, it looks very, very different, actually. So that tells us that there's many, many ways to reconstruct this average data. So many, many different structures which taken together fulfill the population average data. So you would expect like a lot of modes if you take the approach you proposed. And I can hardly imagine that you would be able to distinguish any of them.
Interesting.
Yeah.
I think we're running out of time here, but is there anything else you'd like to bring up?
So definitely one thing regarding my research, our population-based method or multi-state model-based method actually only assumes that the input matrix you get somehow is proportional to a contact probability matrix. That means that you could use the method we developed also on different kinds of data, right? So in the meanwhile, there's alternatives to high C, which are not ligation-based, thus eliminating a big potential for bias and errors. And you could as well use our method on these kinds of data. And you could also furthermore envision to use these multi-state models not only on genome organization problems, but also on, say, quantitative cross-linking to an extent to that's possible. So that would be an important thing to note. And other than that, career-wise for me, I am looking for all kinds of data scientist positions and absolutely would not mind staying in the sort of life science or biomedical field. So if any of your listeners, have an idea, maybe an interesting company I could apply to, I'd be very happy to hear about that.
Absolutely. And you'll be able to find Simeon's contact or some kind of link. You have a website, don't you? So we'll link to your website.
Okay, great.
On the episode page.
Sure.
So people can easily find you. Simeon, this is a very interesting conversation. Thank you for coming on the podcast.
Thank you. You're welcome.